Welcome to NPTEL Inline Certification course on Pharmacognosy and Metabolic Engineering. This is lecture 28, where I will discuss the missing enzymes of vindoline biosynthetic pathway. This is based on one of the latest discoveries made with this biosynthetic pathway. So, what I am going to cover first is the revision of the established route to terpenoid indole alkaloid biosynthesis. So, we will recapitulate the pathway again. Then we will talk about further clarification of T3 oxidase and T3 reductase in vindoline formation, and then we will talk about the biosynthetic steps to tabarsonin and catharanthine formation in Catharanthus roseus. So, let us go to the slide. So, this is what I have covered in one of the previous classes, where I have shown you that tabarsonin converted to vindoline through multiple step reactions, sequentially formation of 16 hydroxy tabarsonin, then 16 methoxy tabarsonin, and then it forms 16 methoxy 2,3 dihydro 3 hydroxy tabarsonin, and this step requires two enzyme activities. One is tabarsonin 3 oxidase and another in tabarsonin 3 reductase, and then subsequently other steps like NMT, D4H and D8 it formed. So, what I would like to talk now is about this step. That means, this issues related to T3O and T3R activities that contributing to vindoline formation. So, 16 methoxy tabarsonin okay. So, let me put it in this way: tabarsonin and 16 methoxy tabarsonin, and in between of course 16 hydroxy tabarsonin is there, but that I am not showing. If you say, I can add it also, no problem. That is 16 hydroxy tabarsonin. So, the enzymes are T16H, then second one is T16OMT. So, this is also now called T16H2. Now, what we have discussed in the last class is this, that these 16 methoxy tabarsonin converted into three hydroxy 16 methoxy 2,3 dihydro tabarsonin, and this was subsequently converted into des-acetoxytabersonin. What color I will use? I will use this color, des-acetoxytabersonin. So, the enzyme which is T3O or T3R, O here is oxidase, R here is reductase. Now, T3O and T3R, so they, if they work separately, then the step cannot be forwarded towards the formation of des-acetoxytabersonin. Okay. Suppose if it happens like that, that out of this oxidase and reductase one of them is not functional, in that case what will happen, that this may leads to formation of (3-hydroxy) to 3-dihydro tabersonin, and this, the next step, it forms des-acetoxy vindorocidin, and this by the action of D4H and DAT. Okay, the space is less, I need to give less space. This will be vindorosin, actually there is a mistake here. Diacetyl vindorosin, and finally it forms vindorosin, VIN, vin, DOROSINE, by the action of DAT, D4H. So, here what happens, what I mean to say, that only T3O is active. So, therefore, in order to make tabersonin, in order to make vindoline, so I put vindoline in this color. These two enzymes will work, have to work these two enzymes have to work together. Okay. So, that what I have mentioned, that it should remain in the form of a metabolon. So, therefore, one product one it catalyzes one reaction, immediately it catalyzes the other reaction. So, otherwise what will happen, that normally if this enzyme T3 oxidase is used separately and a substrate is going is it is if the reaction is catalyzed by if a in vitro reaction can be carried out, in vitro reaction can be carried out with T3O. So, it will never produce a proper substrate for T3R. I think so. If T3O reaction can be carried out in vitro, then the product of T3O cannot be used as a substrate for T3R. So, that is what the point is. So, therefore, T3O and T3R are working as a group and therefore, ultimately it forms, as a result it forms this product. And what will happen if T3O is only working, T3R is not there, then it leads to the formation of vindorosine, not vindoline. That is what I am telling you. Okay, that is for your information. Now, for your information also, that this T3O is basically a cytochrome P450 enzyme, and the T3R is basically an alcohol dehydrogenase. So, the message again I write, product of T3O is an epoxide and are not used by T3R. Therefore, coupled action is required in order to have this activity. So, this was discovered in the year 2015 and the paper published in a famous journal called Proceedings of the National Academy of Science, or PNAS. volume 112, page number 62242, 6229, and this is the first author is Michael et al. And the group leader is Vincenzo Di Luca from Canada. So, this is basically the question which I said at the beginning, that is clarification about the T3O and T3R in vindoline formation. Now, I go to the missing step aspects, that is biosynthetic steps to tabarsonin and catharanthine formation in Catharanthus roseus. So, what we know from the biosynthetic steps to tabarsonin and catharanthine formation in Catharanthus roseus. Okay. So, if we write the step, then the strictosidine glycone it forms gaseosidine, it forms this 421 dehydro gaseosidine, and from here different pathway originates. One route goes to agmalicin and serpentine, and the other route which we are now concerned with, that this makes a product called stemadenine, STEMMADENINE. So, this stemadenine, so from stemadenine to taparsonin or to catharanthine formation, it was not clear, but the organic chemists, they have synthetically shown such reactions. So, but that actually helped now the combinatorial chemist as well as the biologist or the synthetic biologist to work out the step. So, I will not narrate the discovery of this, rather I will just simply go through the steps. So, stemadenine forms okay, because of space shortage, I will put the stemadenine here in the, I put the stemadenine here to start with, STEMMADENINE. This stemadenine will be converted to stemadenine acetate. Stemadenine acetate, that subsequently converted to pre-condylocarpine acetate. This is an unstable compound, and this leads to the formation of dihydro pre-condylocarpine acetate. And that subsequently leads to formation of dihydrosecoidine. Where I will put it? I will put it okay here. Dihydrosecoidine, and from dihydrosecoidine either catharanthine or tabarsonin is formed. Now, if I put the arrow, now the enzyme discovered I will show here. So, stemadenine acetate to pre-condylocarpine acetate, this is PAS, and this one is DPAS, okay, and this is CS TS, it releases acetate away, and then so PA stands for pre-condylocarpine acetate synthase, and DPAS stands for same dihydro pre-condylocarpine synthase. So, a scientist from led by Sarah Connor, that time she was with she was working at John Innes Center, UK. So, they collaborated with several institutions in Germany as well as in UK, other institute like Oxford, and they have elucidated this pathway. So, what they basically identified? They identified two missing enzymes necessary for vinblastine biosynthesis. So, first one is basically an oxidase and a reductase that isomerize stemadenine acetate into dihydro pre-condylocarpine acetate, which is then deacetoxylated and cyclized to form either catharanthine or tabarsonin via two hydrolases. So, so that means the so this is the basically these are the these two new enzymes. So, these two new enzymes they have characterized for the first time from Catharanthus roseus. So, in order to check the activity, so they have done virus induced gene silencing of TS stands for tabarsonin synthase and CS stands for catharanthine synthase. So, when they did virus induced gene silencing, this resulted in marked reduction of tabarsonin as well as catharanthine level goes down, that is one thing. Now, another thing what they have done in order to confirm this function of these enzymes. So, they reconstituted this tabarsonin and catharanthine pathway in Nicotiana benthamina. So, I just go to the next slide because otherwise, so reconstitution of tabarsonin and catharanthine in Nicotiana menthamiana. So, that means these genes they have expressed. So, let me try to show it in this way. So, here the genes for PAS plus D plus CS. So, these three genes were inserted and they add stemadenin acetate here, they fed. And as a result of that, if it can produce the catharanthine, that will indicate that indeed these genes are functional. Similarly, for the other one, what they have done, they made the construct with PAS plus DPAS plus TS. They again do feeding of stemadenin acetate, and that led to the formation of tabarsonin. So, through this they have confirmed that indeed these are the missing enzymes of the pathway, and this is the best way to confirm the function of the newly discovered gene in a heterologous system. So, the tobacco, this pathway is not operative. So, therefore, they have tailored these three genes together, stitched together and put it in the Nicotiana benthamiana plant, and then they have injected in the leaf stemadenin acetate, and as I have shown in the previous slide, that stemadenin acetate will be taken up by the PAS and it forms the precondylocarpine acetate. If PAS is functional there, then it makes precondylocarpine acetate, then DPAS will come and that makes dihydro precondylocarpine acetate, and then similarly the catharanthine synthase or tabarsonin synthase through hydrolase activity, it makes this either tabarsonin or catharanthine. So, similar experiments they carried out here and the results were confirmed. So, the pathway showed how plants create chemical diversity, and this also enabled the development of a heterologous platform for generation of stemadenin derived bioactive compounds. So, stemadenin derived, always problem with my handwriting. Stamandinin derived bioactive compounds can be generated. So, this paper published in the world leading journal. So, one is Nature, another one is Science. I have mentioned this is published in Science in the year of 2018, volume 360, page number 1235 to 1239. So, the first author was Caputi et al. Caputi, Caputi et al. And the group leader, two group leader, one of them is Vincent Coudavalt, French, from Tours, France, and other one is basically Sarah E. Connor. From this So, this is indeed this appears to be very simple, but this is indeed a very complex research, and if you get a chance to read the science paper with care and attention, you will enjoy how they have carried out the experiments, and through these experiments they have confirmed the pathway, and with this the total pathway of vinblastine biosynthesis from tryptamine and cycloguanine is more or less resolved. So, the interesting point is this, that at some later stage the whole pathway may be expressed in yeast system and de novo alkaloid production can be achieved, what was possible in case of tropane alkaloids or isoquinoline alkaloids, which I told you in a few previous classes. So, with this I end this class. Thank you.